make sure you are trying to do that. Now, this one says figure 8 shows a method of producing plants. So, step 1, you can read through that plant. This is a parent plant. Cell picked, taken from it, placed in agar medium, then develop into a tiny plantlet, this one. Then, afterwards, we have plantlets grown in compost. So, what's the question? Some cells in each plantlet develop into root cells. Name the process occurring as these cells develop into. Okay, so what process makes these cells to grow into plants that have different parts? So because it's just one single cell here, they're now developed to have, the plant's going to have root, stem, leaf, so to say. So what's that process? It's just one particular name if you have done your cell biology very well. So the answer to that question is differentiation. Oops, I wasn't supposed to show that part. Differentiation, all right? The next question, let me remind you, differentiation is a process whereby cells that were initially having the same structural identity, some just become unique to be able to carry out a particular function, all right? So describe the advantage of producing plants by this method. Well, you want to remember, what type of method is this? You picked a cell, you didn't have two gametes. Of course, that is asexual reproduction. So, it is faster as it is what? Asexual reproduction. Many plants are produced, their desired characteristics can be retained. What does that mean? Let's say that plant is tall, all of the plantlets will be tall also. But if it was sexual reproduction, when you have a tall plant and a short plant, it is possible you might be lucky to have all offspring tall plants, or some of them may be short or in between. But when it's asexual reproduction, everything you have in the parent will be replicated in the offspring, as it were. Now, an autoclave is used to prepare the agar growth medium used in step 2. So it's talking about these. Autoclave, I believe you know what autoclave is. Autoclave is a sterilizing machine, so to say. So it says here, explain why the agar growth medium is autoclaved. I think I already mentioned the answer already. I say it's a sterilizing machine. So it helps to kill microorganisms. But let's put that in perspective here. To sterilize the agar growth medium, to help to destroy microbes locking on the equipment, because it is possible, in science, you don't want to give chances to any bias. So some microorganisms might interfere with the growth of those plantlets and all of that. So it says, so the microbe does not compete for the nutrient. Can you see that? Because the microbe can take part of the nutrient that should be available to the plantlet, as it were. All right. Next question says, give one of the plantlets had different colored leaves. Give one reason why this plant had different colored leaves, even though they were taken from. Why would one be different? Well, since they were taken from the same plant. What's your guess? It must have been due to what? Mutation, because mutation means a rearrangement in the genetic material of an organism, which could be due to various factors. But once there's a rearrangement, which is, sometimes we say it's abnormal, sometimes we say it's abnormal, but either way, once there's an arrangement in the normal genetic makeup of an organism, that's mutations. Because that's what can be responsible for that kind of thing, because they were all supposed to be the same, but for that one to be different, that means something happened while it was growing, and that is mutation. All right, so the next question here says, crop provide a source of energy in the form of carbohydrate such as starch and sugar. Describe the test to identify starch. This is very simple. If you can also try to watch my video on test for starch. This is about maybe 2 or 3 minutes, very short video. So let's see the answer here. Add iodine solution to the starch sample. If the starch is present, it will turn blue-black. All right, moving on. Next question here says that the amount of energy in the sugar extracted from crop plants can be measured using a calorimeter, all right, shown in this picture here. Let me zoom that bit. So, explain why the calorimeter has a lead? I mean, why does it have this? Is this needed or not? Think. It's the 2-marker question. So the reason is because to prevent heat loss. Now, so when you prevent heat loss, of course, and that's also to prevent water loss by evaporation. So the energy from the sugar is effective enough to heat the water, because what you want to do is that you want the the sugar here is going to be there's going to be respiration, there's going to be heat. So you want all the heat to be retained here. But if you open it up, then the heat will not be retained. So the effect of the heat from the sugar will not be able to go around there, which is not what we really want. So the lid is there to cover it, to prevent heat loss, to prevent water loss also by evaporation, and to retain the heat in that calorimetry so that it can be effective enough. All right. Next question says that state why it is important to stir the water in the calorimeter. So why do you have to keep stirring with this stirrer, as it were? So let's see the answer here. So, to evenly distribute the heat, basically, so that if the heat was around here initially, by the time you stir it, at least it's going to go around, so to say. If you were putting this on a heating thing, maybe we don't have to heat it, stir it, but now that it's not there, you have to distribute the heat evenly, as it were. Moving on, Streptococcus bacteria can cause sore throat or skin infection. An illness called scarlet fever can also develop during an infection with this bacterium, that's Streptococcus bacteria. Give 2 precautions a doctor should take when treating a patient who is infected. Please don't forget this. Already infected. What precautions should he or she take? Let's take a look at this. Okay. He should avoid direct contact if possible. He should use gloves if you have to touch the patient. If you talk about so that infection and response, communicable diseases, the way you prevent. So he should wear protective nose mask. All right. So that when the patient sneezes or anything, it won't get to him. Wash hands after contact. Then sterilize every equipment that was used after the consultation. So while he was attending to the patient, anything he used should be sterilized so that it won't transfer that to the next patient or even to himself, as it were. So it says here, from September 2013 to March 2014, there were
2,830 cases of scarlet fever. From September 2014 to March 2015, there were 5,000 cases of scarlet fever. That means that increase. Calculate the percentage increase in the number of cases of scarlet fever between the periods of September 2014 to March 2015 and September 2013 to March 2014, as it were.
All right. So what we were supposed to do here is just all about the recent value minus the initial value. What's the recent value? This is the recent value. And what's the initial value? This is the initial value minus initial value. Over initial value multiplied by 100, and this is what gives us. So 3, if you minus this, is going to give you this, and this initial value times 100. There you have the increase rate is 110%. That's not nice, sincerely. All right. Now, next question here. Figure 10 shows some Streptococcus bacteria. All right. Some bacteria are motile, meaning they can move themselves. Why is Streptococcus bacterium not motile? It's simple. One of the options here is the answer. Because it does not have flagella. Flagella is that if you draw a bacterium cell like this, it has this thing with which it uses to swim like that and move anywhere it needs to move to. All right. Now, patients with scarlet fever can be treated with antibiotics. Now, new antibiotics need to be tested before they can be used in patients, which is the correct sequence for development of the new medicine. You can remember drug development and discovery. That's what you need to answer this question here. So let's see the answer we have here. So, number 1, you test the drug on tissues and cells. Then you look for testing in unhealthy individuals. So this is called preclinical trial testing, while these is preclinical, so it's called clinical trials. So here you use people that are healthy to see the effect of the drug on these people. And you now look at, you go to double-blind trial, whereby the drug will be given to patients. The drug you're trying to test will be given to patients that have the sickness, and a drug that does not, placebo, which is not a drug, will be given to patients also that have the effect. So we want to know the effect of the, just I don't know if I've made a video on this really, but maybe I'll try and do that after this class so that I can see. There's something called double-blind trial, which means that the doctor and the patient does not know. So let me explain real quick. We have two groups of patients like this. Patient group A, patient group B. They both have the disease. So they have scarlet fever. So you will give the real drug you're trying to test. So real drug goes to one group. The fake, not a fake drug, but just what's it called? Placebo. Placebo is just a drug. No, it's not a drug. It's like a sugar coating. It will have a shape of a drug, but it's usually used. It just, I can't go into explain that special right now. But it's not a real drug. It's just like a pill that has nothing, and it is to test the effect of the mind on drug or on a disease, so to say. But don't worry about that. But the truth is that this is why it's called double-blind trial. So why it's called double-blind trial is because none of these patients knows who is receiving the real drug or the placebo, and the doctor does not know. And the reason why this is being done is that so that the patients that receive the real drug will not fake healing, and the one that does not receive will not also fake healing. So nobody knows who is receiving the real drug. Not even the doctor also knows. All right, so that made sense. That's a quick overview of what double-blind trial means, in case you have to explain that. So moving on. Most cases of scarlet fever occur in children. Adults have usually developed immunity to a toxin that the streptococcus bacteria produce during infection. Explain how an adult develops immunity to toxin. You're just meant to explain immunity, or how you develop immunity against diseases, so to say. So what do? When an adult is exposed to the toxin or the antigen, the toxin or the antigen is what's going to stimulate antibodies. So this stimulates the immune response. This leads to production of what? The B lymphocyte, which eventually produces antibodies, which are in small quantity at first. Please don't forget that. At first, for the first time. Then also, it also produces memory cells are also produced. Now, so these will remember this antigen if the pathogen enters the body again. So what does that mean? So the first time, the B lymphocyte has produced a lot of antibodies. Beautiful. But the moment, few antibodies. But if that disease comes, if that's the pathogen comes in again, then within few seconds or few hours, there will be a lot of antibodies that will produce against scarlet fever specifically. Why? Because memory cells remember what it looks like. So it's going to quickly stimulate the cells of the body that they should attack the cell, as it were. The next one here says that in 2012, two scientists were awarded the Nobel Prize for their research on stem cells. They showed that adult stem cells could be reprogrammed to become cells with properties of embryonic stem cells. Describe the possible benefits of this research. You know what stem cell means? Stem cells are cells that can form any type of specialized cells. So say, for example, somebody has a brain damage, and some cells in the brain, in that part of the brain, has been destroyed. So if you put a stem cell here, it will form the stem cells of that brain part. It's going to form cells of the right brain part. If it's, let's say, cells of the cheek, anywhere you put stem cells, it's going to form that kind of cell. So what is the advantage of doing such thing? Especially using cells from adults now, not from embryo, because there's a lot of contention about using embryo, because it's like you're killing a human being to kill another human. Those are the issues on that. Well, let's see how you do this. What's the advantage? The benefit is this can be used to produce any desired cell type. So say, for example, if somebody has issue with his pancreas, so you can use it to create pancreatic cells that will produce insulin. Now, healthy cells can be used from patients to test themselves, to treat themselves. Beautiful. So you just take, somebody has issues in his body, so you take cell from his own body to cure himself. You program those stem cells, those cells to become stem cells to form the cell you desired on that person's body without having to kill anybody or destroy anybody. So this reduces the chances of cell rejection. Yeah, because that's another thing. Even when you take you transplant organ, there's always an issue of, okay, the person's body will reject the person's antibodies, or immune system will reject the person's organ or cell, so to say. But your own body cells cannot reject your cell. Your immune system cannot reject your cells. So that's why it's reduced that. And so this can be used to treat some incurable diseases. So it's just 3 markables. I think I added 4 points here, which is very good. All right, hope this is making sense. Please do remember to subscribe and share this to your loved ones, please. All right, so that others can also benefit from this. And also, of course, click the notification button so that you can get notified when I upload videos, which I will be doing that a lot more during these few days. All right, so it says here, which is the correct order? Sorry, which is the correct order for the stages? So we have okay, just look at this. Try to attempt this. Pause the video. Try to attempt this. This is the answer we have here. Okay, so it's going to be B. Let's try and label this. So B, R is the first one. R represents prophase. Q represents metaphase. All right, while S represents anaphase. All right, P represents telophase. Because the thing is always written as PMAT. So that's the stages of cells of mitosis and meiosis. So prophase, metaphase, anaphase, telophase. Don't forget that sequence. This arrangement is very good, really. All right. But if it was interphase is not written here. You just see some coily slots here because the chromosomes will not be visible. That time it is called chromatin thread. So this is interphase. If you say something like this, but we call this prophase because it has become visible. If a cell is during its interphase, you can't see the chromosome distinct. It's going to be quite rough like this as it were. All right. So let's go back to our question. So the stage, okay, I already explained this. This should be easy. And I said the stage of mitosis label S is what? S. I just said that some few seconds ago. Try to remember. That's what? Anaphase. I said that some minutes ago from the other one when I was explaining. All right, let me show you to be double sure. So this is what I told you here. Yeah, all right. Moving on. Interphase. Oh, I didn't even know this. Here, the genetic material are replicated. That's what really happens during interphase. Cytoplasm volume also increases. There's growth of cell size. That's what happens during interphase. Then some cell organelles replicate too, like mitochondria will become doubled as it were. But genetic material. So what does that mean? If the cell, like in human, if the cell has 46 chromosomes during that period, it's going to become what's it called, 92. I wrote 46 again because it's going to be another set of 46, so that's going to be 92 altogether, so to say. Please remember, you don't use the word doubles. It's wrong. It doesn't attract mark. You're going to be ignored for that. So the best word to use is duplicate or replicate. All right, moving on. So figure 12 shows a root tip with cell in different stages of mitosis. The image was magnified 400 times. Okay. Explain how a magnification can be obtained using the lens on light microscope. So how do we get this? What are the lenses that were put together to have achieved this? So let's see this here. So this is what happened. So looking through the eyepiece, most of the eyepieces we have through microscope are always times 10 already. So, and usually we used to have objective lens. Please check my video on microscopy, electron and light microscopy on YouTube here. Just check through. So there are different objective lenses. So that objective lens that must have been used to get 400 times will have been 40, because that's going to be 10 times 40. That gives us 400. So the one on the eyepiece is always constantly 10. So multiply that by the 1, because that objective lens is always, some of them can be times 40, times 60, times 100, and it depends, so to say. All right, hope that makes sense. So it is the objective lens and multiplied by the eyepiece lens, which is always already multiplied by times 10. But this one can be times 40, times 60. But for us here, we pick times 40 because that's what looks logically correct here. If this one says times 60 times, that means the one they used was times 60. Oh, that makes sense. All right, moving on. Now, figure 13 shows the pentadactyl limb of a bat and a cat. Describe the reason why the anatomy of the pentadactyl limb suggests that bat and cat evolved from the same common ancestor. All right, structure. Is there another structure there? But don't worry about that, please. Now, describe the reason why anatomy of the... Okay, so why does this happen? That's because it is due to similar or same bone structure, and they have common ancestor that share the same body part. So that means that, I don't want to go into that. If you study evolution very well, you remember that organisms led to each other. So, evolve from each other. So that means that structural resemblance can tell you that their forefathers or four ancestors had the same body parts. In fact, you can say that means they lived a bit of in the same kind of habitat, so to say. But don't worry, this answer is enough for this question. Genetic analysis also provides evidence for evolution. Scientists can sequence genes from different organisms. Describe how this type of genetic analysis helps evidence for evolution. So how does this analysis help us to know that it's evolution really? Okay, it compares the gene sequence from different organisms. So when you look at, for example, now let me give this quick example. A woman was found dead in her room for 2 months. She has been dead for 2 months. Of course, after 2 months, all the flesh had gone off the bones, so they just had some few tissues left. So they took the DNA from these tissues, these bones or whatever it is, and went to test it. And from the sequence, they could tell that that woman was from Caribbean. Now listen to this. How did they know? Because they have the genetic material or sequence of people from Caribbean stored up. So they just compared that. So when it matched people from Caribbean, so they could tell that her origin was from the Caribbean. All right. So it says it compares genetic from different organisms. So you can tell to know this one's from this one from that, so to say. Similar organisms will have identical sequence in their gene, like I told you here. They know that woman was from Caribbean because the genetic mapping matches what was already in the database for Caribbeans. Now, while different organisms will have different genetic sequences, it's that simple. So you can tell. That's what paleontologists use to know that, okay, these organisms were from this, because nobody was there when the organisms were not there. But from genetic sequencing of different organisms, you can tell, okay, this bone resembles this bone, so that's how you can link them all together. All right. Now, Mendel's research on pea plants show that genetic traits are inherited. Which term is used to describe the expression of trait in an organism? That would be what? Mendel research, okay, that was going to be what? It says, describe the expression, please. Expression. That would be phenotype. But if the question says, which of the following can be used to talk about the genetic material or genetic makeup of the organism, that's going to be genotype. All right? Or genome. This, another word for genotype would be genome. Genome, as it were. All right? Hope that made sense. Now, moving on, this makeup is called genotype. So Mendel crossed pea plant that produce round seed with pea plants that produce wrinkle seed. All right? So it's crossing round seed plants with wrinkle seed plants. So all offspring produced were all round seed. Okay. He then crossed these offspring with each other. So he crossed the round seed with round seed, of which they were produced from one round seed and one wrinkle seed. So some pea plants in the next generation produce round seeds and other produce. So when these were crossed, some produce round seed, some produce wrinkle seed. Explain how this showed that some inherited traits are not expressed in the organism. But this is all about talking about heterogeneous, homogeneous, heterozygous, homozygous, then recessive trait and dominant trait. Let's see how that is written here. So all the offspring produced at the first filial generation. What's the first filial generation? The ones that were produced here after you crossed the round seed with, when you cross the round seed with the wrinkle seed. All the offspring produced here are called first filial generation. Now, so the wrinkle seed are recessive while the round seeds are dominant, of course. Now, at second filial generation, the two alleles needed for wrinkle seed were present to be dominant. Please listen to this. What does that even mean? Anytime you have alleles that are recessive means that it is present, but it's not going to be expressive. It's only going to be phenotypically expressive. For example, let me give you this instance. Let's imagine that my mom is short and my dad is tall. Now, and some of them made me. So the gene or the allele for shortness is in me. But of course, I came out to be tall. You can tell from my fingers. Now, that means that the allele for being short is in me, but it's not dominant. It is recessive. However, say for example, my wife has the allele for also being short, which is recessive in her. If I combine my allele for being short plus her allele for being short, we'll give birth to a child that's going to be physically short. Because anytime you have a recessive allele, when the allele combines, then you're going to have a trait that's going to be phenotypically present. So he says here, so the wrinkle seed are recessive while the round seed are dominant. Work with me. So a second filial regeneration. So what it means is that all this is first filial generation. We have 4 offspring. All of them are round seed, but they all have the trait of being wrinkled in them. So the ones that were produced that had wrinkle seed at the second filial generation, it means that there must have been a place whereby 2 wrinkle allele combined to make it show. Because dominant allele, recessive allele will not show when there's a dominant allele. It's only show when there's 2 of them present. So it says second filial generation, 2 alleles needed for wrinkle seed was present to be dominant. So them were present together, so that's why the organism was dominant. So 75% of one of the offspring have wrinkle seed. If I cross that for you here, that would take our time, really. Don't want to do that. I hope you understand. But let me try to do it real quick. The other question, okay, so it doesn't talk about it. Let me try and do that here real quick. First generation, all of the offspring, let's just use letter R for round, small r for being wrinkled. So first generation, this is what happened. All of them were, so round plant was crossed with wrinkled plant. So this is round, this is wrinkled. So when we cross, if we cross all of this out, it's going to give us big R, small r, big R, big R, small r, big R, small r. So you can achieve the same thing with Punnett square. So what it's telling you here is that they now took two of these. So this is this. So this guy is going to be round. Why is it round? Because this guy is dominant, which is round. So this guy is also dominant. This is round. But even though they have recessive trait of being wrinkled there, but it's not going to be shown because this has overrode this. So when you now combine that, so you can do this with your Punnett square too. Big R, big R, big R, small r, big R, small r, small r, small r. So these three, sincerely, phenotypically, will all be round, but this one's going to be wrinkled because two alleles that are recessive have now combined, so it's going to be wrinkled like phenotypically. Hope that made sense. Moving on, I've explained this. Duchenne muscular dystrophy is a recessive sex-linked genetic disorder. All right, this causes muscle weakness. Please, you have to study this. Now, this is unaffected female. This is affected. So this is unaffected female. This is unaffected male. This is a carrier female. This is a affected male. Please, at this point, you have to hear what I'm about to say. This is a very sex-linked traits. Usually, for female, they can either be of these three. It's a law, you have to remember. They can either be unaffected, carrier, or unaffected carrier. Usually, they always most times they always either. Sorry, let me rephrase that. They always usually either carrier or unaffected. But males, they can't be carriers. That they have it, which means they're affected or unaffected. Why? Please hear this clearly. Most sex-linked traits are on X chromosome. So female have two X chromosomes, male have one X chromosome, Y chromosome. So when a female has a sex-linked trait which is on one of her X chromosomes, we say she's a carrier. So this second normal chromosome will help to cover up, so she will not manifest the disease. It will not show in her because this will cover up for the disease. So female are homozygous. So the normal chromosome will cover up for the defective one. But for male, I beg your pardon, when this chromosome is when this chromosome is not affected, there's nothing. Y is not same as X, so it cannot cover for this. So the person will have the disease. That's why females can be carrier. They will have the disease, but it's not showing them because most sex things are on X chromosome, and there's a normal one that can help them to cover up. But for a female to have it, the two X chromosomes must be affected. But for a male, once one X chromosome is affected, he doesn't have any other X chromosome to cover up, so he's going to have the disease all the way. Hope that made sense. You can rewind the video to rewatch that vital information. So let's see what happened here. The person, so there's somebody here that got married to this person. So state and explain the phenotype of person Z, which is this person. Of course, going to be a male. So he is unaffected for the following reason. So how do we know he's unaffected? You can tell from their children. Let's see why. Now, sex-linked traits are always on X chromosome. I just explained that some few minutes ago. And male have one X chromosome and one Y chromosome. So work with me. So only one X chromosome, we say that. So for this reason, he gave his unaffected allele to his daughter. So, because he is X and Y, this woman is X and X, but one of the chromosomes is affected. So see what happened. So he gave his own X to his daughter. So, and the woman gave one of the normal X to his daughter too, to her daughter. So this one will have normal XX, not is affected, but this one is going to get, this is a female, he got normal X from the father, but the defective X from the mother, so she's a carrier. This boy got the Y from the father, got the normal X from the mother, but got Y from, this boy got normal Y from the father, but got defective X from the mother. And I told you, male can either be affected or unaffected. This guy is going to have the disease because he only has one X chromosome. That one X chromosome will be affected. But this lady is a carrier because she has a normal X from her father, which will help her to cover up. Please do rewind this video to watch that part again. It's very important. All right. Oh, sorry for the mix-up at some point there. So to see what is here. So this is a carrier, normal female, affected male, normal male. The reason why this is a normal male is because he got the normal X from the mother. Let me tell you this here. Men will always give their X to the male. That's the law. And we will receive our X from men will always give their X to their female child and give their Y to their male child. It's a law. I say that again. Men or males give their X chromosome to their female child and give their Y chromosome to their male child. While the female will give, of course, we always give X. So we give X to the Y to make a female, to make a male. We'll add the X to the X to make a female. Hope this made sense. All right, moving on. So that was quite elaborate, but I think it was